So I'm going to have you turn to that second section. So chapter 15 is adding on a lot of things that in addition to just regular straight up genetics that Mendel talked about with pea plants. So chapter 14 we learned about dominant recessive, monohybrid traits and crosses and dihybrid and so on, and that's classic genetics. Chapter 15 starts to look, we're looking at some variation upon classic genetics, and so some different things that are going on. And so we learned about this guy named Morgan who, instead of like Mendel crossing pea plants, he crossed fruit flies and looked at traits in fruit flies. And he discovered also with some of that there some certain traits are sex-linked. And so we've been doing, yesterday you did the problems with the sex-linked traits and so on, you just turn those in. And so that's a little bit different, and they're inherited a little bit different than the autosomal traits, and it makes a difference if you're male or female. And then we looked at that couple things with the gender, that in females with the sex chromosome have two X's, and that there is what's called X inactivation, where one X randomly becomes inactive. So that's unique, and I use the example of tortoiseshell or calico cats as an example of that. Last night in the video we talked about how some disorders makes a difference. Two people or two individuals can have the same genotype but have different phenotypes. One may show a disorder and one may not. Based upon what? Does anybody remember from last night's video?
What was your question?
I'll go back over. All right, so it's kind of long-winded here. So last night I talked about in the video that you have two individuals that have the same genotype but exhibit different phenotypes, and how is that possible? What's the reasoning from the video? Isabel?
Wasn't it because it was on the dad's and it only showed up if it was on the dad's?
That's right. You have two of every kind of chromosome. One that you got from dad, that's called the paternal chromosome. One that you get from mom, that's called the maternal chromosome. In some instances, one of the two chromosomes becomes inactivated. And so I used an example with the mice, and so it was the dad's chromosome that becomes inactivated. So whatever gene is on the dad's chromosome, you don't see, and you only see what's on mom's chromosome. So even though you may be, let's say, big R, little r, mom gave you the big R, dad gave you the little r, dad's chromosome becomes inactivated, only the big R shows. But if we flip that and dad gave the big R and mom gave the little r, dad's chromosome becomes inactivated, and now you have this little r, and now you have whatever disorder that is. And so therefore, the two people that have that are big R, little r, their genotype is the same, but yet they exhibit either a normal phenotype or a mutant phenotype, depending upon that. And so then, and then one other thing, just to pull the whole chapter together, is I talked about how a lot of disorders are caused by mutations, like the little r. So you have little r, and you have cystic fibrosis. All right, you have a big R, you don't. So that's a mutation in the gene. But then also genetic disorders can be caused by abnormal numbers of chromosomes. Can somebody give me an example in humans of that?
Isn't it on chromosome 21, if there's a trisomy, then humans can have Down syndrome?
That's correct. Can somebody else tell me, how is it that you'd get a trisomy, which means in that case, three chromosome 21s? How does somebody end up with that? They have two of every kind of chromosome besides 21. Now they have 3 of chromosome number 21, so they'd have a total of 47 chromosomes. Megan. That's correct. All right, so normal production of Agar sperm cells, remember your two pairs of chromosomes, normally they separate. and then they separate again, and so at the end of meiosis, you get one of every kind of chromosome in your egg and sperm cells. So if those original pairs of chromosomes don't separate and they both go to one cell, so then you have a cell that's missing the chromosome and a cell that has two of those chromosomes, and so then it can go to make egg and sperm cells that have two chromosome number 21s, and therefore missing or having an extra piece of DNA there, and then you have certain symptoms. Chromosome 21, if you remember, a karyotype goes from chromosome pair number 1 all the way up to 22, and then the sex chromosomes are chromosome pair number 23. 21 is a really small chromosome because they range them based upon size. So the very, very small chromosome is one of the only trisomies that is viable or humans can survive with. So, for instance, if you had three chromosome number 1s, number 1 is the largest chromosome; it has the most DNA. We don't survive with that extra piece of DNA. So we're very sensitive to how much DNA we have. All right. But trisomy 21 is one of those that we can't survive. Question?
Plants are generally less sensitive to humans, correct?
What was that?
Plants are generally less sensitive.
Yes, plants, we'll talk a little bit more about that later, but plants can survive. I talked in the notes about polyploidy. Plants can survive with more than 2 sets of chromosomes. So I think I used the example of bananas in the notes that they actually have 3. They're 3N. They have 3 sets. So if you did a karyotype of a banana, there'd be 3 chromosome number 1s, 3 chromosome number 2s, and so on. So it's a little unique there. All right, so abnormal numbers of chromosomes, and the last thing that you can do is the structure of the chromosomes can be different. So this is where I went over deletions, where people can have totally missing pieces of DNA of their chromosome, or translocation, where we take one part of the chromosome and attach it to another kind of chromosome, and so on. And so that screws up kind of the order of the bases of the DNA, and therefore what proteins you can make. So that's kind of the gist of chapter 15 so far. So all of these different kind of unique things that deal with genetics. And this last concept with chapter 15 is in the second section, and it's about linked genes. And Morgan discovered linked genes as well, and when he was crossing fruit flies. So we're going to do a few things with this. So each chromosome, if you recall, is made out of one piece of DNA and can have hundreds or thousands of genes. So what does that mean? So let's say we have chromosome pair number 1. Right? And so this is piece of DNA. Remember, a gene is a segment of DNA. So this one piece of DNA can have hundreds or thousands of genes depending upon how long the piece of DNA is. And what does a gene do? Codes for making a protein. And that protein is what gives you a trait. And so therefore each piece of DNA or each chromosome can code for hundreds or thousands of traits. And so that's what that's saying. So then, the genes that are located on the same chromosome tend to be inherited together, and we call those linked genes. What we have been talking about so far, we've done dihybrid crosses. When we did a dihybrid cross, we focused and assuming that the traits were unlinked. What does that mean, unlinked? Well, if they're linked, they're on the same chromosome. So unlinked, they're not on the same chromosome. So when we did dihybrid problems, for instance, like here's chromosome pair number 1. If you're big R, little r here, and let's say that for another trait, let's say this is chromosome pair number 2, they're big N, little n. So the R's and the N's, those genes are on 2 pairs of chromosomes. And so that makes a difference, the unlinked, because when you go to make egg or sperm cells and therefore have kids, you're going to give one chromosome number 1 and one chromosome number 2 to your kids. And so, we're going to see how this is different than being linked. If they were linked, what would be true about these guys? How would I draw them?
The N and the R would be on the same chromosome.
The N and the R would be on the same chromosome, all right? And so therefore, this is unlinked.
For linked genes, can you pair I don't know, that's kind of a dumb question, but I was going to say, can you pair like a dominant N with a recessive R?
Yes. So yeah, that's the thing with this. I'll just tell you, I'm gonna make it like this. But Madeline's question was, could this be this? Absolutely. So without being told some information about it, you wouldn't know.
Okay.
All right, yeah, okay? So linked is on the same chromosome. So what I'd like to do is look at the sheet, before we go on in our notes, look at the sheet you picked up, and look at the side that's at the top says unlinked genes. It gives a definition, unlinked, because this is what we've talked about so far. So it says unlinked genes, and I give you a definition. It says 2 genes located on different chromosome pairs. So that's like the top here. All right, then what I did is I gave you 2 traits. The trait on the left, and I used Morgan's actual traits in notation that he used. So remember, he did something a little bit different in his notation. So if you notice on the left, there's B plus stands for gray body and B stands for black body. So our first trait is body color. And then we have on the right, VG plus is normal wings, VG is vestigial. Remember, vestigial, short, stubby little wings, they can't really fly, all right? And so the second trait is the wing type. So we have body type and body color and wing type. Can somebody remind me what the plus, what did the plus mean when Morgan used that plus? Right, Spencer?
The wild type.
Yeah, the wild type. Can somebody tell me what wild type means? It's not that they're crazy flies. What does it mean? It's the type that shows up the most. It's the most common. And so for a fruit fly, having a gray body and normal wings are the wild type. The other 2, black body and vestigial, are what are called the mutant phenotypes. The reason why they say that is because they are assuming that at one time, for let's say the body color, you have this gray body gene, and all the flies have gray bodies. And in some fly, at some point, that gray body gene got mutated. What does that mean? The order of A's, T's, C's, and G's got changed in some manner. And so instead of making a protein to make gray bodies, now they make a protein that gives them black bodies. So therefore, it's a mutant phenotype due to a mutation of that original gene. And so the same thing goes with normal. So, so the plus does not, though, mean dominant. And this is one thing I'm not a big fan about Morgan's notation, is because he doesn't have anything in his notation to show what's dominant. So looking at this, you have no idea what's dominant. We cannot assume that wild type is always dominant. All right, just because, remember, the 5 fingers or 6 fingers, just because something's dominant doesn't necessarily mean it's more common. So in this case, I'm going to tell you which one's dominant, and that is, in this case, both the wild types are dominant. So gray and normal are dominant. But I just wanted to point out that just because it's that case here doesn't necessarily mean it's always that case. So in just a second here, I'm going to have you go through and do a first cross here. So I give you the 2 flies he crossed. This is the actual crosses Morgan did, and figure out what kind of kids that they could have. But before you get too engrossed in that, let's look at the last thing where it says do a test cross with the offspring of the first cross and figure out the phenotypic ratio you would expect. I want to make sure everybody remembers what a test cross is. We talked about this in chapter 14. Anybody remember what a test cross is? Test cross. Lucas?
Is that where you cross it with both recessive, or cross it just 2 recessive individuals?
Yes. The test cross is usually performed when an individual has the dominant trait, and when it has the dominant trait, you don't know if their second gene is a big R or a little r. So a test cross is performed because so we don't know if they're big R, or big R, little r. We don't know. So what you do is you cross it with a recessive individual, homozygous recessive individual, and you get, if the individual is big R, no matter how many kids you have, you'll never get a recessive kid. So Mendel did this like with peas, and because peas you get hundreds and hundreds of kids. If you never get any, if you have two parents, you do this test cross, and you have 500 kids, and not one of them showed up as little r, most likely that parent has a second big R. But look at theoretically, if they're big R, little r, theoretically you'd get 50-50 showing the recessive and the dominant. And so it doesn't necessarily mean you're going to get exactly 50-50, but if you get any recessives at all, that tells you what the second gene is. And so therefore, a test cross is crossing a dominant individual with a homozygous recessive individual. All right? And so that's what you're going to need to do for that. So I'm going to have you do this first side with unlinked genes right now.